Welcome back to part 2 of neurobiology. In this section, we're going to look at the basic component of the neurological system, the neuron. We're going to look at the neurological developmental processes. Then we're going to talk about the types of receptors that are involved in our medications. We will next talk about the function of synapses and the neurotransmitters and receptors, and then we will end up talking about how synaptic activity is terminated. It will be best if you are viewing this presentation in slide presentation mode so that you can see the change in these blue slides that I use. These blue slides are actually from Stephen Stahl's work. They are the graphics that are used in his book Essentials Psychopharmacology by Stephen Stahl, and they're better if you see them in PowerPoint mode. Let's go on to slide number 2. As I mentioned in our first part, the brain contains over 100 billion neurons that are, as we'll see, weeded out, eliminated, and nourished from trillions that are initially formed between conception and birth. We have over 2 million miles worth of neuronal wires. And each of these 100 billion neurons can be connected to at least 10,000 other neurons. So this gives us a firing and electrical potential of trillions of possibilities. Remember that from our neuronal firing, neurons that fire together wire together and habits are formed. Now, the basic component of the cellular unit is called the neuron, and the neuron has 3 components. It has the soma, which contains the nucleus. It has dendrites, which are projections leading into the soma, and it has axons, which are projections leading away from the soma. For those of you who are not familiar with this type of, think of your hand. Extend your hand, and with your forearm upward perpendicular. Now, your fingers would be the dendrites, and you see how they flow into the palm of your hand. That would be the soma. Then leading out of the palm of your hand and down is one long forearm or one long axon. The neurons have multiple dendrites but only one axon, and the axon ends, let's say, at your elbow, and that would be the axon membrane, and it's called the presynaptic membrane. Now, if you're a contortionist, you can then put your other hand underneath your elbow, and there are the dendrites facing into that synapse, and then going into the soma, down the forearm, boom, boom, connected, all right? Okay, now, neurogenesis is the constant and continual reshaping and remodeling of neurons. As we'll see in our next slide, the process begins at conception and really has a lot of activity up until birth, and then it kind of slows down, but then between ages 6 up into teenage years it flourishes again, and then it settles back down. But our brain is constantly being able to regenerate and renew itself. These neurons can be negatively affected by stress, by aging, by disease, but they can also be positively affected by learning, by exercise, by psychotherapy, by psychopharmacology in adult life. So this neurogenesis occurs primarily in that little part of the brain we call the hippocampus, if you remember that. And finally, glial cells, all of this change and ability to develop is called brain plasticity. And finally, the glial cells are these, I compare them to the packing peanuts you get in a package that protect things. So glial cells are there to protect the neurons. Please continue to use the brain at McGill to explore these concepts. It helps me to understand this process of nerves forming. It's called neurogenesis, the birth of neurons. If I think of pioneer times, so in pioneer times, if we're going to settle a new country, there are first only 1 or 2 pioneers who come out into that area, and then after they have scouted out a new region, others will come and join them. So in utero, The greatest amount of development happens there, but it will continue throughout the lifespan. Nerves are formed from stem cells, and then they can be selected as good potential. And then once they're selected, as we'll see in the next couple of slides, they migrate into the region that they're going to settle, and then they differentiate which functions they will have. And finally, once they have differentiated, they need to build a way to communicate with each other, and that is primarily through the synapses. So we have synaptogenesis. And the majority of this work is taking place in the brain through the second trimester, and then from birth to 5, the brain is going to reach about 95% of its brain volume. But remember, this continues throughout life, primarily in the hippocampus dendrite gyrus of the brain, which is just part of the hippocampus, and that is why we say that there is plasticity of the brain. Now, once the pioneers have settled, they also then have to communicate with each other, and so they start to build ways to communicate, and those are through synapses, so the birth, genesis of synapses. And we have many different ways that synapses are placed. We have presynaptic, which is on the axon, so at the base of an axon is a synaptic membrane, and then the dendrites, remember your fingers and your elbows, at the dendrites is postsynaptic, but there can also be many other areas in which the synapses are formed. On the axon from the dendrite, from the axon to the soma, from the axon to an axon itself. It gets a bit confusing, but just be aware of this pioneer and settler image, and that primarily it is from dendrite to axon, axon to dendrite. The next couple of slides, if you're in the PowerPoint slide presentation mode, will show you what happens in neurodevelopment. You'll notice down at the bottom there are time frames from conception to birth to death, and then the next one over will be conception to birth, and at the bottom just keep following those time frames. But you'll notice on the far left, from conception to birth, is neurogenesis, and that is the forming of those neurons. You'll see that they come out of the stem cell, and they are influenced by a DISC1 enzyme, which stands for disrupted in schizophrenia. But then you'll notice that there are immature neurons, those three little blue figures that look like, I don't know, burrs. And some of them are eliminated and some of them are selected. So you'll see down at the bottom the word selection. So some of those immature neurons are now selected. And then after that, they move over, and other enzymes, an ERB4 and an NRG3, please do not get all tied up in trying to memorize these different enzymes. Just be aware that this is a really complex process, but they are migrating over. And then from conception to birth, you'll see that there is differentiation is the next process taking place. And with the differentiation is myelinization, which is putting this white fatty sheath around the neuron, especially the axon, to facilitate the electrical activity. And then synaptogenesis takes place. And as after synaptogenesis, there is continuing activity through all these different enzymes. You'll notice that from age 6 to 16, there is another flourish of activity where there's again a competitive elimination of neurons and the genesis of new neurons. Oh, that's a lot of information very quickly. Okay, let's move on. Years ago, when we did not know as much as we do today, we thought that possibly there might be some type of gene that caused mental illness. However, that has been greatly disproven. Genes do not encode for mental illnesses, behaviors, or personalities. Instead, they only encode proteins. But in some cases, the genes may produce genetically altered proteins that then will code for subtle molecular abnormalities, which then in turn, from those molecular abnormalities, may be linked to development of psychotic or psychiatric symptoms. Thus, a gene may code for an abnormality in the neurodevelopmental process, or in the synthesis, or of synapses, or in the activity of enzymes, or their transporters, or the receptors, or in, as we'll see in a few moments, signal transduction, all sorts of possibilities. So you'll see on the left that there's this hypothetical gene for a subtle molecular abnormality, and it can take place in any number of areas. You'll see the first arrow going up is neurodevelopment, and then over that it could be something in, as we'll see in the future, the transporters for the neurotransmitters serotonin, dopamine, epinephrine, and GABA, or it could be something in the synthesis of the axon and the dendrites themselves and their own internal transport system of mitochondria, etc. Or it could be, if you're following down under signal transduction pathways, we're going to look at that in a few more slides. This is a very complex process and things could go wrong anywhere in there, or going down farther in synaptic plasticity machinery, or even worse, there could be something wrong at the bottom with the enzymes, the monoamine enzyme and the catalo-methyltransferase COMT enzyme. So lots of things that could go wrong. In psychiatry and mental health, we also talk about nature versus nurture, and this is true. That we were just looking at a slide that talked about all the biochemical, the neurobiological developmental processes that take place, and all the areas along the way where there could be some subtle coding for an abnormal protein that's going to lead to a risk factor. But it isn't just the neurological problems that are here over on the left top of this slide with EAO activator risk gene, or the dysbindin risk gene, or the DISC1 disrupted in schizophrenia 1, or the NRG risk gene. Please, again, you do not have to memorize these. You might be a little curious what the MTHFR one stands for. When that first came up on a screen at a national convention, there was a little gasp and then a lot of tittering, but it doesn't stand for what it sounds like. It stands for a methyl tetrahydrofolate reductase gene, all right? And the COMT is the catalo-methyltransferase gene that is involved with dopamine, as we'll see in a few minutes. So we have all of those. But then over on the right side, you'll see multiple life stressors, and these then, both the risk gene, the factors in the risks of the genes, or in multiple life events, can either stress a circuit, hypoactivating it or overactivating it. And when that happens, the risk genes that are there can then cause the problem. So it isn't just that we have the risk genes, it's also that the life events may then precipitate or activate an underlying weakness or problem that was there. That's called the stress diathesis model, and it figures a lot in schizophrenia, where a person does well up until they're about 17 or 18 years of age, when life stresses begin to increase. They're going off to college or beginning to work, and boom, they have their first onset of psychoses. Now, the way I have found it helpful to remember all of this is that I used to work as a priest in the Dominican Republic, and we were building a central main church out of concrete, which was the main material that we used. And the building was built, and now we were ready to put a roof on it. So first they had to build a subroof of wood as a supporting structure, and there were lots of wood and plywood platforms, etc. And then on top of that, the cement would be poured. Everything was going really well. We were about an hour away from being finished. They were pouring the very last section, and we heard this crack somewhere inside on one of the pieces of support board. There had been some type of internal weakness in that board that nobody could see or know about, and it cracked. And as it cracked, the load of the heavy cement began to open up a hole, and it just got wider and wider. That's my image to help me understand this nurture-nature thing, that the wood had some type of risk factor in it, like we see on the left in this slide with all those genes, and then the continuing pressure of the cement that came on is like the life events, and it was just too much, and the system could not hold, and so it cracked. Let's do a little review before I take you through some more pretty pictures. So we now know that the space between axon and dendrite, or dendrite and axon, is called the cleft, the synaptic cleft. We have a presynaptic membrane, which is on the side of the axon, and in that axon is a vesicle or a sac that is going to contain the different neurotransmitters we'll be talking about in a few minutes. Down the axon, from the soma, down the axon, comes an electrical charge of calcium that's going to hit that vesicle sac and cause what's called exocytosis, which just means getting out of the little sac. And that's going to release neurotransmitters into the synapse that are going to then bind on the postsynaptic membrane. And these neurotransmitters are called first messengers. As they bind into the postsynaptic membrane, there are receptors, many different receptors, not specific necessarily for one transmitter. And those receptors are of several types. They are ion channels that are more rapid and immediate, responding to calcium and sodium, etc. And there are G protein receptors, which are slower, or they're called metabotrophic, and the ion channels are called ionotrophic. So the neurotransmitters enter into these receptors, and the receptors then become activated, and they send out second messengers and third and fourth and fifth messengers, as we'll see in a few minutes. So we're going to be talking about the receptors in greater depth with each mood disorder and major class of medications we study throughout the program. Please remember, you do not have to absorb it all today. These are concepts that we'll be repeating over and over throughout our courses. If this looks like some type of an electrical wiring diagram, it could be, but this is the general structure of a neuron. At the top, you'll see there are dendrites, and those dendrites all lead into that tiny little blue circle there up at the top called the cell body or the soma. And then you see leading out of that cell body or soma this long piece of wire that is the axon, and then you'll see that it finally comes out into presynaptic axon terminals. This is an artist's conception of the genetic structure of a neuron. All neurons are set up to structurally send and receive information. Neurons send information via an axon that forms these presynaptic terminals as the axon passes en passant, passing through, or as the axon ends. This reviews and repeats a little bit of what we've already been talking about, but here you can see it more visually. If you notice up at the top on the left-hand side, it says dendritic tree. Now, that means all the different dendrites that are flowing into into the soma area. And these dendritic trees can take various shapes, and in Stahl's book, he goes into a lot of detail about a pyramidal, a basket, all sorts of different shapes. Please do not go trying to memorize all of those. But those dendritic projections have little spines on them, and in those spines from a synaptic vesicle is where the synaptic vesicle releasing a neurotransmitter can deposit the neurotransmitter. Then if you go down, you'll see that there is the phrase axodendritic synapse. So that's where the axon and the dendrite are coming together in a synapse. But these axons, there can also be an axonal synapse. That's the next one down. Quite frankly, again, all of this is not stuff that we need to memorize. We just need to be aware that there are many different configurations in which there are synapses, not just between a dendrite and an axon, but also between the axon and the axon itself, and between an axon and the soma itself, so many different ways. This is where we see it starting to come together now, the classic way of synaptic neurotransmission or fast communication. So at the top, we have the dendrites, and the dendrites have already received some type of transmission. So there is a reception, you see over in the left-hand column. There has been the reception of some type of message, whether it be on the right-hand side a hormone or a drug or a light or a neurotransmitter or a nerve impulse. Any of these can be received and activate. Then from the dendrite on the right-hand side, you'll see the dendrite. There are lines going into A, which is the soma area. And there in the soma area, there's the integration and chemical encoding. And then on the left-hand side, you see electrical encoding taking place. And then if you go down where it starts to turn yellowish orange, that is the axon. And an electrical impulse is now sent down the axon. And when it gets down to the end of the axon into this area where the vesicles are that we mentioned that contain the neurotransmitters, the electrical impulse activates the vesicle. It's going to open up exocytosis and release, in our case with medications, a neurotransmitter that goes into another dendrite in cell B, and the whole process continues and on down the line. It's hard to put all of this together in a really logical, chronological fashion. We're going to do a little bit of an insert here. You've now seen how an axon communicates with an electrical impulse, releasing a neurotransmitter into the dendrites. It is in that communication between the axon and the dendrite in that synapse that a lot of the activity of the medications that we use in psychiatry and mental health take place. We're going to see in a few minutes that there are what we call transporter cells that take a drug from the synapse, or I'm sorry, take a neurotransmitter from the synapse back up into the axon. And these are what are traditionally called reuptake inhibitors. We'll be talking about this more. Just get the concept that there are transporters that take a neurotransmitter from the synapse back up into the axon. Or there are also receptors that are called G protein linked, and at the bottom of the slide you see that blue with a 7 on it. It's because there is a 7 transmembrane region. Transmembrane means it's going from outside the cell membrane through the cell membrane to the other side. So about 30% of our psychotropic medications work on this G-coupled protein. We're going to look at that again in a moment. Or 30% work on the transmembrane transporters. We'll look again at that in a moment. Once a neurotransmitter is in the synapse, the activity of it has to be terminated in some way. One that we just saw was that they are taken up by those transporters back into the axon. But another way is there are enzymes, and so this little E in the star-shaped kind of figure over here, 10% of our medications work by enzymatically biodegrading the neurotransmitter. The primary one would be our monoamase enzymes. Then there are other ways, though. Down here at the bottom, it looks like a drum. There are 4 transmembrane regions that are ligand-gated ion channels, and that just means that a neurotransmitter such as GABA, which we'll talk about again in a moment, will bind into the top of that drum, and about 20% of our medications work that way. And finally, over here on the far right-hand side, there are 6 transmembrane region receptors, and these are voltage gated, or ion, meaning sodium and calcium are the primary ones, and 10% of our psychotropic medications work that way. Let's look at one of the main concepts of the way our medications work, and that is through what's called a G protein linked system. Look at all the different components on this slide, and then in the next few slides you're going to see some changes. So at the very top we have a first messenger, which would be a neurotransmitter, dopamine, serotonin, norepinephrine, etc. And it's going to come down, and you notice in this blue design there's kind of a V shaped at the top, and if you'll notice, it matches the V shape of the first messenger, the slightly green up above. All right, now if you follow to the bottom of this blue figure with the 7 on it, you see this jagged edge. Well, when the first messenger at the top binds into that V, watch the change of that jagged part at the bottom of this 7 receptor. And then underneath it we have a slightly different color blue. This is the G protein. It is going to be bound, and you notice it looks kind of like a whale, but the tail of the whale is going to change shape so that it can bind to this pinkish enzyme over on the right. In this slide you see that the neurotransmitter has bound, so there it is at the top, bound and linked. And now if you go down to the bottom of that 7 greenish figure, then you see that now it is no longer jagged, but it has rounded out, and it is able to configure to the G protein below it. Next slide. Now the G protein and the 7 membrane areas have joined together, and watch what's going to happen over here in between the G protein and the enzyme. It is changing shape so it can bind to the enzyme, and once it binds to the enzyme, then there are going to be a whole bunch of other activities taking place, and this is called signal transduction cascade. What happens from the neurotransmitter binding, starting this G protein, binding to an enzyme, the enzyme initiating another activity that leads to a third, a fourth, and a fifth. Now over here at the top, you see what we have just looked at in the previous slides. The first messenger neurotransmitter bound to the 7 membrane receptor, and then the G protein linked up to it, and that bound then to an enzyme. Over here then at the top right, the enzyme is bound to the G-coupled protein, and it is now sending out a second messenger, and this second messenger is going to go down and bind to other enzymes, whether to activate them or to inactivate them. On the left-hand side, they have become inactivated. On the right-hand side, they have become activated. If they become activated, then further signal transductions are going to take place. So all of this is quite complicated. We probably will never need to know exactly what's going on in order to practice and prescribe. However, this whole signal transduction cascade process does not take place instantaneously. It can take time, and that is why our medications do not work. Most of them do not work overnight. We continue on from the first messenger into the second messenger. If the second messenger activates a kinase enzyme, that's going to then activate or inactivate a fourth messenger. It's called phosphoprotein. Or the first messenger doesn't necessarily have to be a neurotransmitter. Over on the right-hand side, you see the first messenger might actually be an enzyme, as the second messenger coming down and activating a third messenger, which is a phosphatase instead of a kinase on the left-hand side. Again, please, you do not have to know all of this. It's just to make you aware that when we are giving a medication that is activating or increasing the presence or decreasing the presence of a neurotransmitter, we are setting into motion a whole cascade of events in the neurological system. But it is this downstream, further at the bottom here, you see it says fourth messenger activation, and then diverse biological responses. Well, some of those responses could be the activation of new genes or the inactivation of genes. One more slide on this, and please don't panic, but I just want you to see in a visual how all of this can kind of come together. So at the top, we see a G protein linked neurotransmitter. We also have an ion channel linked neurotransmitter. Going over a little bit more to the right, we have a hormone, and then on the far right, we have a neurotropin. All of those seem to be at the top of like a glass or a tube. Well, that top is the synapse and the membrane. We're coming into the synapse, and now we are going into the postsynaptic membrane. And if you follow down on the left-hand side, you can see that there's this first messenger, second messenger, third, fourth messenger system, and you can see that different enzymes are involved. And it is very, very complex. If you want to read about this more slowly and find out what all those alphabetical abbreviations stand for, you can go to your Stahl Chapter 1 and look up the slide or the blue graphic 11, and then from there you can read about all of these things. But please, you don't need to memorize or remember this complexity. It is just to make you aware that there is a lot going on when a person swallows a pill. Okay, we're almost at the end of this section. One more blue slide, and that is to show what I've been saying, that our pills and medications do not work overnight. There is, as you follow from the bottom to the top, binding of a first messenger, the activation of the enzyme ion channels, and then the formation of second messengers, and those second messengers activating the third and fourth messengers. Those third and fourth messengers then activate some early genes, and those early genes then go on to activate the late genes, and then those late genes go on to have long-term effects of starting or stopping processes in our neurobiological systems. And if you go back down to the bottom of this slide, you can see the timeline that some of these things do take place rather quickly, but then others take up to a day to start taking place, and then it can take up to 10 days or more. And that is why many of our antidepressants, we tell clients that it takes anywhere from 2 to 6 weeks for them to take effect, and we'll be seeing why in the next part of our presentation. All right, one more slide to put this all together. We have talked about the brain as an anatomical, electrical, and chemical system, and what we've been looking at in this portion is the anatomical structure of the neuron, then its electrical and chemical activity that take place down the axon into the synapse, and from the synapse into the postsynaptic membranes. So we have that synaptic transition, the release of neurotransmitters through electrical stimuli, then these chemicals that have been released, neurotransmitters, ions, etc., starting other electrical or chemical processes in March. And now let's talk one more concept. We've got the neurotransmitter in this synapse. How do we terminate its activity? There are three main ways. The first is the neurotransmitters are taken back up into the presynaptic membrane. In the next presentation of this whole topic, we'll be looking at the various neurotransmitters. They are primarily 5-HT, which is serotonin, dopamine, which is DA, norepinephrine, which is NE. We'll also be looking at glutamate, and we'll be looking at GABA. These can all be transported back up into the axon and then restored in those vesicles. The next is enzymes in the synaptic cleft. There are enzymes in there that biodegrade, and this can take place through acetylcholine is the neurotransmitter, and it is terminated by acetylcholine esterase, or the cells, the neurotransmitters just kind of wander off and they're absorbed into the glial cells and reprocessed, and acetylcholine is the only one that is handled primarily in this way. So here's my image. Think upon the synapse like Madison Avenue in New York, where there has been a big parade and thousands and hundreds of thousands of people. So and we'll compare the neurotransmitters in the synapse to either the crowd or to the pieces of the ticker tape. So first of all, to get rid of all these people, along come trolleys, and they take people back to where their cars are parked, etc. And that is the reuptake process. It's a trolley that comes along, takes them back to where they belong. Then we have all that stuff still left over, all the trash. That's like the enzymes, and the enzymes are like the street cleaners that come along, shred up what's left, and get rid of it. And then there are still some pieces left over, some people that didn't get on a trolley, some trash that's still left there, and along comes a strong wind and it blows all of that away, and people diffuse off, wander off to find their places, or the trash gets blown into other areas. That's what we call diffusion. So we have reuptake, we have enzymatic degradation, and we have diffusion into the glial cells. All right. Now let's take one more picture to see if we can put this all together. This is a very simple graphic that tries to bring this all together. So you see there this long thing with all sorts of stuff in it. In the center of this graphic, it is the axon. And you see in the axon that there are different molecules. These are the 4 little circle pie slices that are acted upon by enzymes, and those are like the coils that take those little pieces of the pie and they put them together into a neurotransmitter, which is step number 2, and those neurotransmitters then are stored in these in these sacs that have the 3 blue circles in them representing the neurotransmitter. So the circle is the vesicle and the blue is the neurotransmitters that have been formed from precursors that we'll talk about later and enzymes. Now, once the electrical impulse comes down, you'll see here that the neurotransmitters can kind of leak out of their vesicle and they're put back in. But number 4 is that the electrical impulse comes down and it hits the vesicle sac, releasing it. So you'll see down here at the very bottom, this is the presynaptic axon membrane, and there is this kind of cove that has been created, and it is opened up, and you see the little blue ball dropping out. That is the neurotransmitter. This is what's called exocytosis, and the entrance of the molecule, which is the neurotransmitter, into the synaptic cleft. Now, in the synaptic cleft, they are going to then bind either to postsynaptic receptors, which are these little cupped green things at the bottom on the next membrane, which is the postsynaptic membrane. They'll either bind there, or you'll notice that on the presynaptic membrane, there are also some of those little cups, and some of the neurotransmitters, the little blue balls, are binding there. That is autoreceptor, and the autoreceptor's function is to tell the axon, hey, we turn on, turn off. We'll be looking at this a lot more. So there are 7 steps. The neurotransmitter is synthesized, and then it is stored in vesicle sacs, and then some of those will be leaking out and destroyed by various enzymes, but then number 4, action potentials, will cause the vesicle sac to fuse with the membrane and open up and release the neurotransmitter into the synaptic cleft, which is number 5 on the left. And then number 6, the released neurotransmitters will bind to either postsynaptic receptors or to presynaptic receptors. And then finally, number 7, these released neurotransmitters are going to be deactivated in one of 3 ways that we talked about in the previous slide. Either the trolley will take them back up into the axon, or an enzyme in here will destroy them, or they will just kind of diffuse off to either side and be reabsorbed into the glial cells. That is the end of this lecture. And in the next slide, there is a link to go to Quizlet.com. Please take advantage of that. Test yourself, because it is only by having to recall and retrieve this information that you're going to solidify it for yourself. You're going to make those neurons fire together so they wire together and you remember this material.